Okay, it's time. Um, Welcome everyone to the last part of this workshop, and now completely in English. Um, so let's see. We have still a lot of participants, 57 participants. I'm very happy about everyone who showed up. I mean, this is going to be, especially given that you have, or a lot of you have only started with this a couple of weeks ago, or maybe 5-6 weeks ago. This is going to be challenging, but I think it is doable, and I think you're going to learn a lot about the way things, well, the way this works. Okay, so let's get started. I think anyone who still wants to join can do so as we're going along. Also, we are, of course, recording this, so even if you can't follow everything today, you will have the opportunity to go back, rewatch the video, and then follow along, maybe pause it when, in case you have any issues or you're not sure about something, and then go back and keep watching it. So again, I am very grateful for the invitation to be one of the presenters in this workshop. I think this is a great opportunity for people who maybe haven't had the chance to learn these kinds of techniques or these kinds of to get this kind of statistical knowledge in the courses that they took at university, or maybe just aren't sure about how things work, but who would like to, of course, apply them in their research. So I'm going to talk about linear mixed models and, of course, repeated measures designs. And this is really, these are really key designs for linguists, especially for psycholinguists, right? So if we are going to use language, if we're going to study language, then usually, and I'll explain why today, usually what we will want to use are mixed models, right? So, okay, so let's get started. Let's get started. So what do we want to do today? And we're probably not going to get into everything at the start here, or in the first session, but that is fine, because we have a second session where we will talk about some more advanced parts of this and some more practical parts of this. So basically what we will probably get to today is we'll try to apply a linear mixed model to fixation times in a typical eye movement data set and interpret the results. Why are we going to do an eye movement data set? Well, because that's my research and that's one of my data sets. So it's data that I know very well. So for me, it's great to use that as an example. And for you, it's great to have that as, well, to have a real life data set rather than something that was made up. So a real life data set from psychology of language, or you could also call it psycholinguistics, that was actually published and where I show you how I did the analysis. Okay, so some notes about the format. So I've used R and RStudio to actually make these slides. So they're generated using a package called Sharingan. They are made from an R Markdown document, and these R Markdown documents contain both R code and formatted text. And it's really nice if you want to present your results in a way that's transparent and replicable. Because you actually have the R code and you have the data with the slides, and you can recreate everything just from the R Markdown document and from the R code in the slides. So if someone, and if you publish your presentation in that way, I mean, not only does it look nice, at least I think that this format looks quite nice, the included styles, but anyone who wants to know how you came up with the results can just look into the R Markdown document and see exactly what you have done. Okay, so what I would recommend as we go through this, so I'm going to, of course, show you the slides, but I will also show you the R Markdown document that produced the slides. And on the website for this course, you can find the files in the folder with my name, and there you can so you can download them, you can put them on an empty folder in your computer, and then if you double click this R project file, so the Part 5a R project, and have RStudio already installed, which you should have from past sessions, then it should automatically start with that project open. Okay, and I can show you what that will look like. So if I, let me see if that works. So now I think you should be able to see my RStudio. Okay, so someone has problems finding the documents. They were uploaded just now. So if you have another check in the folder, then yeah. So I think you just found them. Yeah. So they might just have appeared there. So so if you look at the project that I opened, right? So you can see this is the R Markdown document that I have, and I'll explain some of these parts of code. But the main thing is that you can see that what's in the slides is all in here in R Markdown, right? And the nice thing is that you can go through, and for example, you have this. For example, we have this bit of R code here that that helps you install any packages that you might need, and also loads the packages that we will need right now. So if you want to load that, you can just click that little green arrow on the top right corner of the code chunk, and then R will just run that. Well, it will run any chunks up to this one and this one. And if you look in the console, then you can see it being run here. Oh, it should be here. Yeah, so you can see it being run here, and then when it has finished running, you can see the output of that command. So here it's just basically saying that there are a few packages that we're attaching that have functions with the same name as packages that were already loaded, but that's not really an issue here, right? So so this is a really nice way for you to follow this presentation. So you can just go through. I'll have the slides up mostly, but I can always switch to our studio. You can see the text of the slides here. And our studio will even show you things like pictures just in line here. So you can really read the slides either in the slide format that I have there. So that would be the HTML file that's in that folder. So the Part 5a presentation .html. I've also made a PDF just in case people have problems opening the HTML file, or you can just open this as the project, open the RMD file, and then you can follow along there and just run each code chunk bit by bit, right? Okay. So, all right. So let's go back to the slides. What's also important, so most of the time, of course, I'll be looking at the slides, but I will also monitor the chat. So if you have any questions, just put them in the chat, and at some point, I hope I will stop and see them, and then we can talk about them. So just put your questions right into the chat. Don't wait. Since you're not interrupting me, so I'll just keep an eye on the chat, and this way I can tell if people have issues following along, or if people need some more time or have some question. And we'll take a couple of breaks to enable people to ask questions and follow along, to make sure that people can follow along properly, because it is going to be new for a lot of people. Okay, so let's go back to the slides. All right, so yeah, so my recommendation is open those files and try to go along, open our project, open the Part 5a presentation.rmd, and try to go along with what I'm doing. So for example, start by, if I'm going back, yeah, I'll just be going back and forth, I believe, between the two. So here, for example, start by installing the packages. So if you remove the little hash mark here and then run the chunk, it will actually download and install the packages that you need. So that could be useful if you don't have them all installed. You don't necessarily need these two for the analyses, but if you wanted to make the slides, you would need those. And yeah, so this is also a chance for me to share how I work, right? How I make slides, how I do this. This is the first time that I'm using these slides for teaching. I've used a different system to make the slides, so I hope everything works out all right. But I just looked over them and they look fine, so that's good. Okay, so going back to the slides now. And here we are. Okay, so yeah, so this is again how you run the code chunks, and what we have in the slides as well is you have the R output, so you can also see the output from the slides here, and you can see the R code that I'm running. And again, in R Studio, the output from the commands appears underneath the code chunk, so that's very useful for you following along. And instead of pressing this little green arrow, you can also press Control, Shift, and R to run each chunk. Okay, so let's talk about the data set that we are going to use today. So this is a data set that from an experiment that was part of my dissertation. So it's by Angele and it's a paper by Angele and Reiner. This is actually, if I can again switch over to our studio here, the paper is in the files that are available. So if I click on that, I can actually open it. And let me just see if I can just share that as well. So here we go. That's this is the paper. So it is published research. What we did in this study is let me see if I can get that here. Yeah, what we did in this study. Screen sharing is paused. So let me see if I can get that back. Okay, there we go. So this was a study about parafoveal processing. So parafoveal processing, when we're talking about reading, means essentially the question: how much information do you get from the upcoming word while you are still looking at the previous word? So we know that skilled readers don't just process the current word, but they also, whenever they can, they start processing the upcoming word. Okay, so and that is one of the things that makes reading really efficient. Okay, so one of the things that isn't really clear is how readers deal with the fact that they have both the information about the current word that they're looking at and possibly information about the upcoming word available at the same time. Now, we know that pre-processing of the upcoming word helps with identifying that word once you look at it. But in this particular experiment, what we wanted to do was determine if having this parafoveal information could possibly also influence processing of the word that you're currently looking at. And the way we did this was like this. So we're actually going to be looking at experiment 2 here, but I'm just going to tell you about the first experiment because it's the same idea for now, and then we'll go to experiment 2. So in this experiment, we use the gaze contingent boundary paradigm, and this is a paradigm that my PhD advisor Keith Rayner came up with, and it enables. So it requires eye tracking, so it needs you to know where the person is looking. And it enables you to manipulate what information about the upcoming word is available while a participant is reading to the left of the word. So basically, the way this works is you have a single sentence on the screen, like this one: Victor read the news once this morning. Okay. somewhere in this sentence, usually between two regions of interest, you put an invisible boundary. So that is indicated by this dashed line here. I can make it a little bit bigger. I can try. There you go. This is indicated by this dashed line here. So this boundary was invisible to participants. Participants didn't know that the boundary was there. But we monitored, so in this experiment, we monitored participants' gaze position, so where they were looking in the sentence. And while participants were looking to the left of the boundary, so while they were looking at Victor read the news, we were able to manipulate what appeared on the screen. And here in experiment 1, that could be either the actual word that was going to follow, like once here, or it could be a repetition of the word to the left of the boundary, so news, or it could be an unrelated word, or it could be a non-word in this particular experiment 1. Okay, so so while participants are looking on this side of the screen, basically, while they're looking to the left of the boundary, in all of these conditions, the word or the letter string that they're seeing to the right of the boundary is different. But they don't. But they, of course, they don't know that it's different, or because they don't know which condition they are in. Now, the really interesting thing comes once participants move their eyes across the boundary. So let's say they're in the repetition preview condition. They're reading here, Victor read the news. Now they're looking at news, and they're having the copy of news to the right in the paraphobia, so to the right of where they're looking. Once participants cross the boundary, the preview that was here on the screen is very quickly replaced by the actual word that's supposed to be in the sentence in this position. So in this case, no matter which condition you were in, once you looked at this word, you would see once, because that's the only continuation of the sentence that makes sense, right? And if you do this correctly, and if you do this quickly enough, then participants will not notice that there was any kind of change on the screen, and they will just keep reading normally and naturally. So the key thing is that the paraphobial preprocessing of these experimentally manipulated previews is not enough to make participants actually consciously notice that there was something else on the screen. It is just enough so that participants, so that you can influence how the word processing and the word identification or the sentence reading processes that the participants are engaging with. So that processing is affected, but not to the point where they would actually notice it. So there will be a difference in fixation time, at least on the post-boundary word, on the pre-boundary word. Okay, so but participants don't notice the manipulation. Okay, so what we actually, so the data set that we're actually going to look at today is going to be from experiment 2. So in experiment 2, we had very similar conditions to experiment 1, but we added a couple of conditions to try to see what was going on with this information. So we had the control preview. Victor read the news once this morning. Then we had the repetition preview. So Victor read the news this morning. And again, people never actually, so once people looked here, they always saw once. So they never actually read news in this position. Just while they were looking here, they might have had a copy of the word they were looking at in the paraphobia. Similarly here, the question here is, if you don't have an exact copy, but you have a similar letter string that only differs by one letter, will that help you? If you have a semantically related preview, will that help you? And then as a control, if you have a preview that is not related to anything, how does that differ from actually having the control preview? And we were mostly interested in how this would affect fixation times on the pre-boundary word. So essentially, does information that appears to the right of where you're looking, does that affect how you process the word that you're currently looking at? Okay. All right. So let's go back to the presentation. And let's see. So here's that figure with the conditions again. And now let's try to read in this data set. So this data set is provided to you. You can read it like this in R, right? And it's in that second code chunk that I've prepared for you in the Markdown file. Okay, so basically we're using the read underscore CSV from tidyverse. And what it does is it gives us an idea of what it's reading the column says and so on. That's not really that interesting. It also says, oh, there was a column name missing, so the column with the observations actually didn't have a name, so it just called it an X1. Otherwise, it's read properly. Now, if we look at the data, so another neat thing about the slides is that you can actually have embed tables and things like that. Here we're just looking at the first 5 entries, excuse me, of that file. So you can see here and I'm also just looking at the first 10 columns in that file. There's a lot more. So here you can see we've got one column that says which subject the observation came from. Then we got one column that says which item the observation came from. So item in this case means sentence. So which sentence was the participant reading, or was the subject reading? And then we have different fixation times. So we have different durations. So here we have first fixation duration on word N. So word N is the word before the boundary. Excuse me. So that is just the way these are named. So word N is the word to the left of the boundary. Word N1 is, or N+1 is the first word after the boundary. Word N+2 is the second word after the boundary. That's how you usually label these in iMovement experiments. And here, so the first couple of conditions that of measures that you're seeing here this is first fixation duration, so abbreviated as FFD. And first fixation duration is the duration of the very first fixation that a participant makes on that particular region. In this case, that region is either the pre-boundary word or the first post-boundary word or the second post-boundary word. So here we have those measures. And then here we have gaze duration, which is another eye movement measure. Gaze duration is the sum of all the fixations that you make on the word before you leave it. So you, because sometimes you refixate a word. Sometimes you make more than one fixation on a word. And in that case, first fixation duration is not any different, right? Because so here in this case, so here we have a case where a person only made one fixation on this region, right? So we have first fixation duration, 232 milliseconds. So these values are in milliseconds. And gaze duration is also 232 milliseconds. So that means that this particular participant only made one fixation on that word, right? Here we have a case where first fixation is 187 milliseconds, but gaze duration is 334 milliseconds. So that means that this participant made more than one fixation on this word in this trial. And that's why gaze duration is higher here than first fixation duration. In all of the other cases here, you don't have any difference between first fixation duration and gaze duration. So that means that for all of these cases, only a single fixation was made, right? And another thing that we notice here about these data, and that will become important later on, is that we have missing values. So look, so here, this person does not have any observation in first fixation duration on the pre-boundary word. On this sentence, they also don't have any measure on here. On these two sentences, they do. And what happens with eye tracking is, of course, you don't control where participants look. Participants control that. So if a participant doesn't look at a word, if they skip the word, then of course, so if they go directly from a word further to the left in the sentence to word n+1, for example, then you're not going to have any observations here. So then you're going to have missing data here. And the data point here is missing simply because the participant didn't look. Right, so it's not missing because of any experimental error. It's just because you don't have because of what the participant did. So this is typical for eye movement data, and for that, linear mixed models are very useful since they provide us with a way to very easily deal with these kinds of missing data. Okay, so. So let's take, so I mean, in the slide, this table is nice, but we can, in our studio, we can take a more detailed look at this. I'm just noticing that we have 99 participants by now, so very happy that so many people are still around with this and still following along. Okay, let's actually look in our studio here. Let's go to the code chunk where we are loading the file. Okay, let's just run it. And so now we have this data frame. In this case, I think it's, yeah, we can call it a data frame, E. And we can, so if we click on it, or if we just write into the console view E, then we can also see what is what is in this data frame. Right, and you can see that after the gaze duration measure, there are a lot more columns here. So we have TVT, which is total viewing time, which is the sum of all the fixations that participants made on this region or on this word, even if they left it to the right and then came back later. And we have single fixation duration. So here in single fixation duration, we only count, we only record a value if really there was only one fixation on this particular word, right, in this particular trial. So here, these NAs mean that there is no data here, and with single fixation duration, I mean, there is no data whenever the participant simply didn't make any fixations or made more than one fixation, right? Okay, so moving on, we have number of fixations, we have whether the word was skipped, and we'll go back to that later. With go past time, maybe we'll have a chance to look at that. And then here we have our condition all the way at the end, which is called preview, and which can be correct, repeated, orthographic, semantic, unrelated. And if you remember, if we're going back, if you remember from the table that we just looked at, those were the preview conditions, right? So this column tells us which was the preview condition that we had here. Okay, and then here, if we look at the different rows, then we have, so here we have our first subject looking at item 21. Here we have our first subject looking at item 22, 23, 24, and so on and so on. If we go far enough down, then at some point we will see our second subject looking at item 1, 2, 3, 4, 5, and so on. And so on. So we have one row for each trial here. And then in the columns, we have the fixation time measures for the different regions. Okay, so just so you know what the structure of this file is that we are going to analyze. Okay, going back to the presentation now. So the first thing that we would like to do, okay, someone's asking, are the items words? Very good question. They are sentences, actually. So if I go back to our studio here, so each item is one sentence. So here, for example, we've got sentence 73, then FFDN is the pre-boundary word in sentence 73. So is the first fixation duration on the pre-boundary word in sentence 73. Then FFDN1 is the first fixation duration on the post-boundary word on sentence 73. So item in this case is sentences. Because in this experimental design that we used for this, each trial was one sentence. Okay, great. So let's go back. And yeah, and I'm really happy that people are asking questions immediately, because that means I can address them. And it also means that people are actually following along, and that people are actually, so that I haven't lost everyone in my explanations yet. Okay. So the first thing, so when you're opening a data file like that, a good idea of what you might want to do is you might want to do some very, very basic check for outliers. So here I've given you a quick list of what all of these measures are. And one thing that we might want to do is just get an idea of what's the mean of these measures? What's the range of these measures? Roughly, maybe what's the distribution of these measures? And for example, do we have single fixations or first fixation, gaze duration? Then we have FMLT, which is first of multiple fixations, which is basically the opposite of single fixation duration. So here, so actually you could say that first fixation duration is single fixation duration plus the first of multiple fixations, and then gaze duration is, of course, single fixation duration plus first of multiple plus second of multiple fixations. But we almost never use FMLT and SMLT. I just have them there for completeness sake. So anyway, when you have measures where you only have a few fixations, one or maybe two or maximum maybe 3 or 4 fixations that go into the measure, then we would want to look for unusual values that are below 80 milliseconds, because no fixation, but if you have fixation durations of under 80 milliseconds, that fixation duration cannot reflect anything about the processing of the word, because usually preparing a saccade, so saccades are the eye movements that you make, the fast eye movements that you make to go from one word to another, and fixations, of course, the time intervals between the saccades. Usually, these saccades take about 100 to 120 milliseconds to prepare in the brain, so or in the oculomotor system. So if you've only if you're making a saccade that fast, so if you land on a word, you stay less than 80 milliseconds, and then immediately you move your eyes, then it's very likely that the eye movement that you're executing has actually started preparing before you even looked at, before you even started fixating on that word, before you got any information from that word, at least any foveal information on that word, so any information from the center of your vision. So there are some things like express saccades, so sometimes fixation of just above 80 milliseconds might still tell us something about the word, but if it's below 80, it's just probably, well, it's very likely that the participant got no information at all from the word that they were fixating, and rather they made a mistake in looking at that word, and they're immediately correcting it. They detected the mistake even before they landed on the word, and then so they land on it and they immediately correct it. So we don't want those kinds of fixations in our mixed in with the fixations where people actually got some useful information from the word, and maybe the fixation duration is influenced by that information. We don't want those fixations mixed in with these kinds of express fixations where they don't, where participants clearly haven't obtained any useful data from the word, right? And then we also don't want fixations over 800 milliseconds, or you could even say 500 milliseconds. I'm being very careful here by saying 800 milliseconds. We don't want these really, really long fixations because it's very, very rare for a person to make these very, very long fixations. Usually, the average fixation duration is, depending on the task, is between 200 milliseconds and, let's say, maximum 400 milliseconds. In reading, it's usually around 250 milliseconds. So if someone is not moving their eyes for almost a second, so for 800 milliseconds or more, there is something going on. I mean, one possibility is they're just staring at the word, they're not actually reading, they could be falling asleep, there could be lots of things going on, but it's not reading. So we also don't want those kinds of fixations in there. And then for measures involving multiple fixations, like total viewing time and go past time, which is the sum of all the fixation durations that you need to go to after you've entered a word to leave a word to the right. There we might consider values above 1,500 quite high. So if we do extensive refixating and extensive sort of rereading, then maybe that's also an issue. So we might also want to look at that. For the other measures, we don't really have comparable rules of thumb. Okay, so how do we do? What would be the easiest sort of one-line way of just getting an idea of sort of the range, mean, standard deviation of these measures? Put it in the chat if you want to. What would that command be? How could we take a look at just that? Just some very simple statistics for all of the columns. No one? Okay. So one very simple way of doing this, and it doesn't show up great. The output doesn't show up great on the slides, but you can, of course, just use the summary command. So the summary gives you an idea of the range, and it doesn't come out very nicely here. It looks nicer when you look at it in our studio. But basically, here we've got, so this column is subject, this one is item, condition, and then here it goes a little bit off the screen. So let's just take a look at it in our studio, actually. So let's just go over here, go down here, and just run this code chunk here, right? And here you've got, so you can see that it's writing the summary in here, and it's a little bit easier to read on here, and it's writing a lot. That's why it's taking a little bit of time, but there we go. So so essentially the first column is just the index of the observation, so that's not particularly interesting. For the second one, well, we see that subject, the minimum is 1, the maximum is 40. So so that means we have 40 subjects in there. Item, the minimum is 1, the maximum is 120. So that's also, I mean, that means we have 120 sentences. Now, let's look at first fixation duration here. So we're looking over here now. You can see we have maximum values. They're all below 800, and minimum values, they're all above 80. The reason for that is that I've actually already filtered that file for these kinds of outliers. But in general, I mean, if you get a new file from someone else, you don't know that. So it might be a good idea to just do a quick summary of the data to see what you're working with. So, and you can see the same thing applies here to total viewing time, to all the measures. For total viewing time, I've thrown out everything that was above 1,500. For the other ones, I've thrown out everything that was above 800. Right, you can see that with the max, or below 80. Okay, so that's our summary. Now let's go back to the presentation and keep going with this. So now let's calculate some means. And there's a slide that wasn't showing up here, so I'm just going to go back to our studio. Maybe it's because we're missing some lines here. Okay, so calculating means. So let's just calculate means over all of the data points, and let's try this. So here in this code, we don't we don't actually get correct, if you use this code, we don't actually get correct values because all of the values are NAs. And the reason why that happens, so if I just run this and then run and then get you to, I'll get it to show you the E dot means, you see that all the results here are NA. Why is that? Why is that the case? Actually, yeah. So why do we just get NAs here? Well, the reason for it is that if we do any kind of calculation with NA, the result is NA. So, for example, if I do in the console, if I do NA plus 1, the result is NA. So a number that we don't know plus 1 is a number that we don't know, right? And in the same way, if I want to calculate the mean of, let's say, 1, 2, 3, I can get that mean. But if as soon as I add NA, the mean is going to be NA, right? There is a way to avoid this. And so this behavior is on purpose so that you don't accidentally compute a mean of a variable that contains missing values without being aware that it contains missing values. But if you know that it contains missing values and you know why the values are missing, for example, just because you're using eye movement data and people just didn't look at that word. So in that case, the missing data points are perfectly explainable, and it's fine to just remove them. Then you can tell the mean command to do exactly that by writing NARM equals true. And then see, then it will just ignore the NA if you give it that option. It will just ignore the NA and give you the same mean as for the 3 values before. Okay, so what we can do here now is we can just use NARM equals true for all of these commands, and then have it show us the means, and there they go. So this is for the post-boundary word SFD N1, and if we wanted to, we could do the same thing for the pre-boundary word SFD N as well. So we would just have to replace all the N1s with N, right? Okay. And then you can take a look at this. Okay, and then we have this as well. And here, so you can see for the pre-boundary word, and this actually, I actually realized now that I'm looking at this, that SFDN is actually the word before the pre-boundary word. So it's the word before news. SFD1 is the word news in the example sentence. So let's put it back. Let's put it back in. Let's look at the word news here, and it's not just news. I mean, it's all of the words. Put that back in. Let's run it again, and you can see here there's the interesting observation in the means, which we will talk about testing later, that people are actually faster at processing the word news, or at least they're making shorter fixation durations on the word news when the preview was either repeated or orthographically related, right? So that is the surprising thing that we found in this study, and you can see it here in the means. Now, okay, now we now there are different ways of doing this to get actually the number of observations right, because we need to because of course we need to account for the fact that some of these conditions, we might have fewer observations. For example, if in some of these conditions people are more likely to skip the post-boundary word or the pre-boundary word in this case, then we would have a lower n here. And so we can either do this by making sure that that we're only considering cases where we don't have an A's here when we calculate the length, because length will also count observations that are an A, or we can just use tidyverse, put a filter in here so only use observations where SFDN1 was not an A, right? And then again, group by preview and then calculate means and so on. So, and if we do that, then again, we get the correct number of observations. Okay, so now we are exploring this data set. So this is just as much a course on or a class on how to deal with new data sets as it is on linear mixed models, but we'll get to the linear mixed models. So let's make a simple linear model. Let's do a simple, maybe not terribly useful analysis, where we just try to predict the fixation duration on word n+1, so the pre-boundary word, from the fixation duration on the preceding word. I don't know if you have any intuitions about what that's going to look like or how that is going to work, but we can of course do it, right? So let's do that here. And you can see from the results of the linear model. So this is not a linear mixed model. This is a simple regression like you have used in the previous sessions. And here, so here we get a significant relationship, or well, we get that the coefficient that predict the coefficient for the predictor SFDN, so for the single fixation time on the preceding word, is significant, and it has a very low p-value. Okay, so and just in terms of what this LM1 object actually is, if we just call it like that, if I just have it, we just have it printed. What's in this object LM1, or in this variable LM1, is actually so the function call, and here the two coefficients, right? Intercept and the coefficient for SFDN. And if you look at it here in the environment window, then you can also see that it has the two coefficients, or 192.8, 0.108. Then it has all the residuals. So it's a list of different things. It has all the it has all the effects. It has all the fitted values and so on. so it stores a lot of information about the model in this variable, and then when we just tell R to print what's in this variable, it prints this, the function call and the coefficients. If we use the function summary on this model variable, then it will print us a more detailed output with the call, with not the residuals, but sort of an indication of what the distribution of the residuals is, and then the coefficient, significant codes, and then the F-test for the model. Okay, and of course, and then so this is just to show you that there are different functions that do different things with the same object. So you can, and this is something that is special to R. So you can have an object or a variable of a certain class. Like if we just check what class, so we can say class lm1, what that is, and it's class lm, right? So so we can have an object or a variable of class lm, and different of these generic functions like plot do different things depending on what class they are asked to work with. So here, this plot function, what it actually does is it gives you a residuals versus fitted plot. And here you can already see what we're going to talk about more a little bit next week, is that these are data that, so fixation durations, just like response times, are distributed with a long right tail. So you can see there the distribution is, so there are lots more observations to the right of what's sort of the median or the mean of this distribution, right, that are to the right than there are to the left. And if we look at the normal QQ plot, we also see here on the right, we see, so the idea behind a QQ plot, right, is that if the so you're plotting these quantiles of your distribution, and if your distribution is a theoretically perfect normal distribution, then your residuals, so if your distribution of the residuals in this case is a perfect normal distribution, then it should follow this diagonal line, so this dashed line here. And here you also see that that there are more data points in these quantiles to the right of the mean, which is 0 here, than there should be. So that is having a sort of a it is having a thick long tail. Okay, and then we have well, scale and location. That's not that interesting. And then residuals versus leverage, where you can maybe identify outliers. Not that interesting, but it's nice that they have it for this this plot. Okay, going forward with this, let's look at the regression equation. And so here, I'm also giving you an example of how to typeset a formula in Markdown. So here you can see I've used these $2 signs to say this is a formula. And RStudio just gives me a preview of what this will look like right in the editor window. So that is quite nice. Right, so, and what I'm, the way I'm writing this here is I'm using the same expression that I would use for documents in LaTeX that might not be very familiar to people, but you can you can look it up easily. So for example, if I want to write alpha, I do a backslash and write alpha, and then it appears as the Greek letter here. Okay, so just as a reminder, the regression equation is we're predicting each observation. So I, in this case, is the index of our observation. So each observation yi, we're predicting with the intercept, which here is alpha, plus plus a coefficient. Plus a coefficient times a predictor, which in this case, our dependent or predicted variable could be the single fixation duration on word n+1, and the predictor could be single fixation on word n, right? And then this coefficient is sort of the scale, or it tells us how strong is the relationship between these two, plus the error. Okay, and you can see that the intercept doesn't have this little index I, because it's always the same for each observation. And the coefficient also doesn't have the little I, because it also is the same for each observation. The predictor variable is, of course, different for each observation, right? Each observation has a different SFD of word n. Of course, all the predicted values, every single predicted value is different, so it also has this here. And then the error is also, of course, different for each observation. Okay, so we can plot the data points along with the regression line. So we can do that here. Oh, and we would actually, oh, I see, would actually have to hold it together. It's nicer to show it in the presentation then. So let's just go forward. We've just looked at all of this in our studio. So, and you can also see how nice this comes out then for rendering the formula. Okay, so here we've got the relationship. So we've got SFDN and SFDN1, and if we plot the regression line, you can see there is a clear positive relationship. So the higher SFDN, the higher SFDN1. It's not a super strong relationship. There's lots of variation, but it's definitely there. Okay, so as a reminder, the way we test this is, the way we test whether this relationship is actually there is, or one of the ways we can use to test this is we can fit a model that only has the intercept. That's what the 1 does here. We can fit a model that has nothing but the intercept, and here's the plot of that model, right? So this model predicts every single observation just using the mean, right? The question is this model, so is this model here worse than the model that we saw here, which actually predicts each observation with the or from the single fixation duration on the preceding word, right? So is there a difference in terms of the variance explained? Okay, so we can test that, and I'm just going to switch back to our studio to show you the issue here. So here we've got the intercept only model, and I'm asking here, in this case, the intercept is the same as a very common descriptor statistic, and of course, if we have to pick one single value to best explain the data with, and we are using the method of least squares. So least squares is how we do regression, right? So how we fit the regression line. We're trying to minimize the squared errors of the observations, right? So in that case, the value that, or the statistic that, the value that gives us the best possible prediction, if we have to use one single value to predict everything, is the mean, right? So in this case, the intercept just reflects the mean. Okay, so, and it's the mean of y, right? So we're predicting every observation, every single fixation duration onward n+1 with the mean of all single fixations onward n+1. Okay, now we can do a model comparison with this command ANOVA. If we try to uncomment this here, well, in this case, LM2 was not found, so I just have to run this first here because I hadn't run this chunk yet, right? And it's going to give us that same output as before, and it's going to give us that plot. Okay, so now it says here that models were not all fitted to the same size of data sets. So this is something that you need to keep in mind when you are comparing different regression models. So you can only compare models that were fitted to the same data sets, okay, or the same size of data set at least. So, and if you look here, if you look up here, so our model LM1, if we look at it where we fitted it here, up here, we fitted it to a subset of data where we don't have any missing data in SFDN, and we don't have any missing data in SFDN1. So this function isNA tells us it's true whenever there is a missing value here, and it's not true when there's not a missing value here. So we can only include here observations that have both a value for n and a value for n1. Otherwise, how are we going to predict the one thing with the other if one of these are missing, or if both of them are missing, right? On the other hand, and actually you don't have to do this exclusion. The LM command will do it automatically. So even if I don't say data equals subset here and have these explicit criteria, LM will automatically exclude any incomplete observations, so where you don't have the predictor and the predicted value, right? And you can also see from the degrees of freedom here that here we have 1,260 observations, right? Now let's look at LM2, the one which was here, the one where we're predicting SFDN1 with the intercept, right? So here, actually, whether we have a missing value in SFDN is not relevant, right? Because we're predicting everything with the mean of SFD1. So, of course, R does not exclude data based on whether there's a missing value in SFDN. And if you look at the degrees of freedom here, you can see that actually a lot more data points are going into this regression analysis, right? So what we need to do is we need to just make sure that both of the regression analyses use the same data set. And the way we can do it is just say for each of these, so we're fitting them again, is just to say for each of these, make sure that you only include data where SFDN is not NA. Otherwise, we can't compare them. And this is how we then get that comparison, right? So, and if you notice, 0.006981, that is the same p-value, if you remember, that we got here, that we got here for in the model where we're trying to predict SFDN1 with SFDN, right? 0.00698. What that means is that essentially doing this t-test to test if the coefficient is 0, or to test the null hypothesis that the coefficient for SFDN is 0, is exactly mathematically equivalent, right, to comparing a model that has the slope for SFDN with a model that has just the intercept, right? So here we've got an F value of 11.551 and a p-value of 0.006981, right? If we look back up here, you can see we have the same F statistic here. It's the exact same test. And another neat little trick that a lot of people don't know is that if we only have one predictor, then we can get the F value by just doing 3.399 squared, right? So here, really, it is the same analysis. The ANOVA here is the same thing as a regression analysis, and that is the case for all ANOVAs and regressions. And when we want to understand how linear mixed models and ANOVA, so how linear mixed models tie in with ANOVAs, then it is very important to understand how these two methods work together, or how they are basically the same thing. Okay, moving on now to thinking about why there's a correlation. I think this is better viewed in the presentation. So let's go there again. So why is there a correlation? Why would there be a correlation between the single fixation duration on one word and the single fixation duration on the next word? And again, we're still not looking at what we find really interesting here. We're just thinking about the way measures work together in a general way. So maybe it's just because both measurements come from the same participant, right? So each time we are looking at SFDN and SFDN1, we are looking at pairs of observations that come from the same participant, because they are reading the same, they're from the same trial, right? So you know that for all of these statistical methods, there is an assumption called the independence assumption. So the independence assumption says that there should not be any systematic relationships between values in the data, except for those that are accounted for in the model. So you should not have unexplained variance, essentially, in the model, at least not systematic unexplained variance. I mean, you will always have unexplained variance, but the idea is, of course, that this unexplained variance is completely random. So it's not correlated, the errors are not correlated with any of the predictors or with the predicted value, right? They are just completely random. And they just reflect things like measurement error or just general variability that all averages out to zero, right? If you test enough participants, or to close to zero. Okay, so if we have a systematic relationship between values and the data that's not accounted for in the model, that is a problem. And of course, I mean, this is called Introduction to Regression Models for linguists with R, right? As being linguists or psycholinguists, we of course have data sets where we have these independence problems all the time, because we don't just usually go and test one person once, right? We go and we have a person in the lab or doing our online study, and while we're having them doing our study, of course we want to get more data than just one observation from them. So we're having them do, I don't know, maybe 100 trials, 120 trials, maybe 200 trials, right? So that means we could have 200 observations or more from a single participant, and we don't know when we are getting the data. We don't know if there is systematic variance from these different participants, or due to these different participants. So if maybe all the values that come from one participant have some systematic thing in common, but it is very easy to imagine why they would. For example, if you have a participant who's tired, they're going to be slower, probably on all of your measures across the experiment, right? So all of the measures that you collected from this one person are going to be a little bit longer than measures that you collect from a different person. Or if the person has just had a coffee before doing your experiment, then maybe they'll be faster on all of the measures, right? So that's a reason why there might be systematic relationships. And then of course you also just have individual differences. So maybe some people just make longer fixation durations in general. Maybe some people make shorter fixation durations in general. And you don't really know if that is the case until you have taken a look at this in your data. But if it's a possibility in your design, then you have to be able to account for it. So the model that we just used, given that we don't know if there are systematic effects of participant, we should not be using it, right? Because if the independence assumption may be violated, then actually we can't interpret the tests. All of the null hypothesis significance tests that we do are dependent on the assumption that there is independence in between the values and the data, except for those that are accounted for in the model. So if we're not accounting for systematic variance by participants, then essentially, even if we get a very small p-value, that very small p-value might not actually be caused by the effect that we're looking for, but rather by systematic differences between participants, which is not what we're looking for, at least not usually, right? And not in this particular experiment. So this is very important. Okay, so how do we account for the variance? Let's try a bad idea. So one bad idea that we could do, or one thing that we could do, is we could simply say, okay, well, I need to account for the variance, for differences between systematic differences between subjects. I know that I can account for things, for variables, by just adding them as a predictor to the model. So here I'm doing that. So I'm just making subject into a factor. So to tell R that that it's actually a categorical variable. So that we have subject 1, subject 2, subject 3, and it's not a continuous variable where 4 is twice as much as 2 and so on. It's just subject 4. That's the fourth subject, right? And I add it to the model as a predictor, and then I get something like this, right? So then I get my my single fixation duration predictor, and interestingly, it's not significant here. And then I get contrasts for so I have 40 participants, right? So I get 39 contrasts, and each of these contrasts tells me whether the average fixation time for that particular subject was different from the baseline, which in this case is just subject 1. So each of these, this one tells me, is there a difference, a systematic difference between subject 1 and subject 2? And here is the systematic difference between subject 1 and subject 3, and so on. Now, of course, and then this, and this goes on up until 40, right? Of course, so what we did here was we added subject as a fixed effect. So this means we have essentially added it as a categorical variable with 40 different levels. And we have these 39 treatment contrasts, each one comparing one subject to the baseline. Is this what we actually want? No, it's not. Because what we're doing here is we are saying we are interested in only these 40 subjects and no one else. And we care about the specific differences between each one. And this is usually what we do when we put together experimental conditions, right? In that case, we are only interested in these, for example, 3 experimental conditions that we have created. And we definitely care about specific differences between each one. For subjects, this is not the case. We really only want to account for the variability that is introduced by the systematic differences between the subjects, and not for the actual differences between each one. We don't care, because the subjects, we're just having the subjects as a random sample from the greater population of subjects. And you can talk about what your population of subject is. Like, is it university students, or is it all people? Or, and if your population is all people, then are university students a good sample from that, and so on. But what all of these arguments have in common is, of course, that we don't really care about subject 1 versus subject 2. We just have subject 1 and subject 2 as samples from a bigger population that we want to make statements about. So we want to generalize to other subjects. And we can't do that if we have subject as a fixed effect. So we need to use random effects. So instead of assuming that there are fixed group levels with one true population mean value for each group level, we assume that the group levels themselves are samples from a larger population. So instead of saying, okay, we have 40 participants, those 40 participants are all we care about, we don't care about anyone else, we're saying our 40 participants are samples from a larger population. So we're not doing the experiment with 40 predetermined subjects. we are randomly choosing the participants that we're going to include from the population of all possible participants. And we only care about these particular participants insofar as they are samples from the population that we care about. That's very different from a fixed effect. And in terms of items, in terms of our language, we can also argue that even though we wrote the sentences that we're going to include, really these sentences are just samples from a much larger population of all possible experimental sentences. So we do not care about the particular experimental sentence or word that we have included in the experiment. We only care about this particular sentence insofar as it is a sample from the larger population of all possible sentences to which we want to generalize. So we can repeat the experiment with different subjects and possibly different sentences. And we will have we will have a different sample there, but they will still be from the same populations. Whereas if we repeat the experiment with different fixed effects, so with different conditions, then it is a different experiment, right? Okay, so let's talk about random effects models. And this is an extension of the regression model that we've talked about before and that you've talked about in previous weeks. So basically, here we now split the intercept up into a general intercept, which is called mu in this case, which is common to all participants and which is a fixed effect, and a random intercept, which is called alpha I. So I is in this case the subject number. So each individual subject gets their own random intercept, which can differ from the overall intercept. And then, of course, we have error. Okay, so, and the idea is that these random intercepts are not completely free to vary. So just now when we did this, when we put in subject as a fixed effect, we put nothing in to constrain the estimates here. So we didn't make any assumptions about, for example, a distribution that these different estimates, these different coefficients for the different subjects would be coming from, right? Here in the random effects model, we do. So we assume that that the random effects, that these random intercepts all come from a normal distribution with a mean of 0 and one particular variance. So instead of trying to fit subject as a fixed factor with 40 different means, we fit it as a random factor and we estimate only the variance, not 40 different factor level means, right? And what this also does is it constrains the random intercepts. It in a way it pulls them together. It pulls the estimates for the subject intercepts together. This is called shrinkage, and the reason why I'll talk about that in a second, but the reason why it's sometimes difficult to estimate the degrees of freedom for a model with random effects is this shrinkage. So the fact that you are not estimating, so you're not estimating 40 different means for 40 different subjects, which would cost you 39 degrees of freedom. Instead, you are just estimating one variance. But that variance is also not exactly just 1 degree of freedom, because it includes so many observations from different subjects. So we have to estimate the number of degrees of freedom, essentially. Okay, let's make the mixed effects model a little bit more complex. Okay, so now we have the intercept, and we have the random effect of subject. So the thing with the random intercept is it's basically the same thing as having a main effect of subject, right? So we have this random intercept. So main effect of subject in this case means that all of the observations from this subject are going to be a little bit slower, a little bit faster. So here we have our overall intercept, which is a fixed effect. Then we have our random intercept, which is a random effect. And now we have a dummy contrast. So now so for example, if we wanted to just compare the identical preview condition with the dissimilar control preview condition, okay, then we would need one contrast, and that contrast we could call Dij. And so in this case, I is participant and J is the number of observation. So for example, we could have I equals 1 and J equals 1. We would have, that would be participant 1, and J would be the observation within that participant, so observation 1. And then the next observation would be 1, 2, would be participant 1, observation 2. We could have participant 2, observation 1, and so on, right? That's the idea behind those indices, right? And so, of course, the dummy contrast is different for each observation. The Y variable is different for each observation, because that's in this case, for example, the single fixation duration that we observe. The error is different between each observation. The overall intercept is the same for each observation. The coefficient here is a fixed effect. It's also the same for each observation. The random intercept, so alpha I here, is the same for each observation within the same participant, right? So it has only I, it doesn't have J. Okay, let's make it even more complex. So there may be an overall effect of preview, but some subjects might show a stronger effect of the repetition preview than others. So it means we need both a fixed and a random effect of that should read preview, and I can fix it directly here in the file of preview. Okay, so since preview has 2 levels, since preview has 5 levels, right? You remember, we need 3 new dummy contrasts. So we had one already. So now we need D2, D3, and D4. So this might look a little bit complicated, okay, but it is just an extension of what we looked at just now. Okay, so we still have our observation, our single fixation duration in this case, which is yij. So it's the jth observation number j for subject number I, right? We still have the intercept overall. We still have the random intercept by subject in this case. Then we have our 4 contrasts, D1, D2, D3, and D4. Because if you remember, you need, we have a factor with 5 levels. And so in order to have this factor with 5 levels in the model, we need 4 degrees of freedom or 4 slopes, 4 different, so 4 different contrasts. And we'll look at this in a second as to what the contrasts actually look like, right? And then we have also 4 fixed coefficients. So this one here is the fixed part. This is the same for every participant, right? The beta 1, beta 2, beta 3, beta 4. Those are the fixed effects. And then, because we also care about, well, we don't necessarily care about, but we need to include the possibility in the model. We need to include this possibility in the model. We have random coefficients as well. So these coefficients, again, have this I index. So that means that they are different for each participant. So participant 1 has can have a different intercept and a different coefficient for this contrast here. And what this means in practice is that, for example, participant 1 is less sensitive to the repetition manipulation than participant 2, right? So maybe participant 2 shows a stronger effect of the repetition than participant 1. In the previous model here, this one, if you imagine all these other contrasts in here, this previous model could not account for that, right? This previous model can only account for what you could think of as the main effect of participants. So it could say, it could account for participant 1 having an overall longer fixation times than subject 2. This model allows us to have participant 1 have not only longer fixation times maybe, but also a stronger or a weaker effect of for example, the repetition. Okay, so we got a question about spontaneous production. I mean, it depends on what you measure. If you measure response times, yes, definitely. I mean, the same person is going to produce words at probably at a similar rate. So probably there is a systematic difference between your first participant and your second participant. Maybe your first participant just produces words faster than the second participant. So yes, so you would have, and then if you have different conditions that also affect response time, those would have an effect. Those you could capture in a fixed effect, and then you could capture the differences between the systematic differences between participants. Like one participant just talks faster than the other participant. You can have that as a random effect. And then, of course, you could also have the possibility that one participant is more affected by your manipulation than another participant. So yes, spontaneous production is also a language experiment. So it has repeated measures, and the same considerations apply. And it's as long as you have something that you can compare between participants and within participants, like response time, you have to take these things into account. Yes, so this random coefficient is essentially an interaction between subject and preview, right? So different subjects here can have different effects of preview. Okay, so how do we actually use models with random effects? Or how do we account for these random effects? The simplest and easiest way to include one random effect in a model is to use a repeated measures analysis of variance. So, and that is what most people have done for the last 40, 50 years, right? 60 years, right? It's because it is also computationally easy to do. You can do this by hand, unlike linear mixed models, which you cannot do by hand. So this is a different kind of analysis from the multiple regression models. Well, from the interpreting slopes, from the t-tests. It is very similar to the f-tests that we've talked about for the multiple regression model. So we are comparing models. And in the simplest case, a one-way repeated measures ANOVA, we compare a model with just the random effect, so just the systematic difference between participants, to a model with the random effect and a fixed effect, right? So we don't need to worry about the random effect of subject. We just eliminate the variance associated with them, and we're done. It doesn't work when we have multiple random factors. So either if they're hierarchical, like students within schools, or crossed, which is what, as language researchers, we have most of the time. Crossed would mean that all subjects see all items, and all items are seen by all subjects, right? And also, you can't include continuous effects into an ANOVA, because in the ANOVA we only deal with categorical effects, and yes, so yes, we would. Okay, so how do we fit a model? Well, the harder way is linear mixed models. And the advantage of the linear mixed models is that we can include as many random factors and effects as we like. And we can also use and interpret contrasts. You've talked about that in previous sessions. LMMs are robust to unbalanced design, so you don't have to worry about things like sphericity violations, which is a problem with repeated measures ANOVAs, but you can't use least squares in linear mixed models. So instead, you have to estimate the model in an iterative way. So, so essentially, you have the computer try lots of different solutions to the equations that you gave the model, right? Every regression analysis, in a way, is solving a set of equations. So in this case, you estimate the model iteratively, and you use, instead of the least squares criterion, so instead of minimizing the squared errors, you try to find the parameter values of the model with the highest likelihood, where likelihood means that so it's essentially given the data, what are the parameters that are most likely to have produced the data, that fit the best with the data. Okay, so in LMMs, you use either maximum likelihood or a variation of maximum likelihood called restricted maximum likelihood. You don't have to worry about the differences between the two. It's just usually people who are interested more in the fixed effects than the random effects use RAML, so restricted maximum likelihood. So I would recommend that you use that too. Okay, and because this is an iterative process, the model can fail to converge if there is not enough data. So, so we'll talk about that a little bit next week, about the considerations, like what do you do when your model doesn't converge. Now, first of all, let's talk about the easy way. So you might remember from previous experience with statistics, paired t-tests, repeated measures, the repeated measures ANOVA allows us to do a similar thing. So in paired t-tests, you just compare pairs of values, and by doing that, you are able to take out all the differences by comparing just differences between conditions, you're able to take out all the systematic effects that are not related to the difference, like overall in some pairs both values are higher than in other pairs. You don't care about that. Because you just care about the difference, you take everything else out. And similarly, for a repeated measures ANOVA, you are able to split the sums of squares. So where sums of squares are essentially an estimate or a function of the total variance. So if you divide the sums of squares by the degrees of freedom, you get the mean squares, which is an estimate of variance, right? So sums of squares total is sums of squares between participants plus sums of squares within participants, right? So, and essentially you're saying we don't care at all about sums of squares between participants, because those are all the differences, systematic differences between participants. We only care about the sums of squares within participants, and so the sums of squares within participants are the sums of squares explained by the model. So those are the fixed effects, and the residual, which is the uncorrelated, so which is the error that is not correlated with participants or is not related to participants, right? So all of those bits that I showed you in the model here before, so the random intercept here, and also the random coefficients here, those all go into the sums of squares between participants, and only the fixed effects here go into the sums of squares model, because they are the ones explained by the model. So by doing that, we get rid of the independence variation if the only random source of variance is subject, in this case, of course. We have less unexplained variance, and as a result, we have higher power. Okay, so just quickly to tell you the way, the notation I'm going to use here, in the regression equations, I've been using I to represent the participant. Here, in this case, in these ANOVA, in this ANOVA notation, I represents the factor level, M represents the participant, and P represents the number of factor levels, N represents the number of participants. So just just to show you how simple it is to calculate this, right? So the total sums of squares, we just sum up, we just subtract the mean from every single observation. So we use the squared differences, so the squared differences from the mean. This is basically a variance estimate, right? For all of these. So that's the total sums of squares. That's our estimate of the total variance. Okay, and we have, and this is the degrees of freedom, which is essentially just the number of observations minus 1. Then for the between participant sums of squares, we just assume that every value is can be explained by the participant mean. So by the mean value for this participant, and so the variance that is explained by the differences between participants is so for each observation. That's why we multiply it with p. We essentially, instead of the actual observation, we just plug in the mean for the participant, for this particular participant, subtract, so get the difference of that from the overall mean and square the differences. Okay, so that gives us the sums of squares between participants. And then for the within participant sums of squares, we, instead of subtracting the overall mean, like we do here, from each individual observation, we now subtract the participant mean from each individual observation. Okay, so so that's how we get those, and then we can from the and then we can of course get the model sums of squares, where we, instead of each individual observation, we put in the model prediction, which for a one-way repeated measures model is just the mean of the factor level therein, and we subtract from that the overall mean. And well, the residual sums of squares is just what's left over when you subtract the sums of squares model from the sums of squares within participant. And then what you get here, so these are just the degrees of freedom, then what you get here is basically you just compare, you get an F value by comparing the mean squares model and the mean squares residual. And the mean squares, of course, the sums of squares divided by the degrees of freedom. Okay, so let's do this. So let's get the preview effect on single fixation duration on word n+1, which is the pre-boundary word. So the first thing we have to do is we have to get a data frame, or we have to get a table where we have one mean per condition per subject. So we have to calculate, so we have to take all the observations, all the individual observations for each subject in one particular condition, and we have to calculate the mean for that. Okay, so here, for example, so we can do this using the tidy words, the dplyr. So we just calculate, put in a new variable called subject means, and we get that by grouping by subject and preview, and then we use summarize with mean and na.rm equals true to get the mean of SFD n1 for each combination of subject and preview. And here is what you get out of that. So we have this is, for example, the first 4 conditions for subject 1 and the condition means for subject 1, and then we have the condition means for subject 2, the condition means for subject 3, and so on. So that's what this looks like, that we've generated, and we can use this this particular table here to do the repeated measures ANOVA. And the way we do that is we can use the library EZ, and which is the easiest way to do that. And from that library, you use the command EZ ANOVA. We just tell it this is the data variable. So this is the table or the data frame that we have our data in. And then we just give it the names of the columns where our different types of data are. So dv is the dependent variable. So we tell it it's that mean. So look here, we have where we have the mean single fixation duration on word N1 for this particular person and in this particular condition, right? WID is the within ID. So this is basically saying, well, what is the random factor? So what are the observations within, right? So this is subject. And then within equals preview as saying, we have, so the actual fixed effect that we want to look at. So subject is the random effect. The actual fixed effect that we want to look at is preview. And then this is what we get. So it turns out, so it does the ANOVA. It also does the sphericity test for us. So in the ANOVA, we have a significant effect of preview, right? We also have a significant test for sphericity. So we would have to correct for this, but both the Greenhouse-Geiser and the Huhn-Feld corrections are significant here. So we would still reject the null hypothesis that preview has no effect on single fixation duration on word N+1, right? And essentially what this F-test does, again, is it compares a model that only has the systematic effect of subject to a model that has both the systematic effect of subject and the systematic fixed effect of preview. Okay, so we could say we are done now, but there is still a potential independence violation, which is items, and we cannot include more than one random effect in a repeated measures ANOVA. It doesn't work. So if you have no other choice but to use an ANOVA, for example, because you don't have a computer, not really a problem anymore, but it was in the past, you can do that, but you have to do one ANOVA for each random effect. So if in our design we have systematic effects of both subjects and items, for example, faces, sentences, in our kind of research, it's almost always items, right? It's almost our second random effect is almost always words, sentences, paragraphs, things like that. Then we have to do a second ANOVA. So the first ANOVA we do over subject means, just like the one we just did. The second ANOVA we do over item means. And the F value from the first ANOVA is usually known as F1, and the F value from the second ANOVA is usually known as F2. So here's how we do the F2 ANOVA. So you can see we do the same, we calculate our item means in the same ways. Actually, if you don't give, easy ANOVA the item means, but you just give it the unaggregated data, it will do this step automatically for you and just give you a warning. But it is nicer to have the control over this too. So to calculate these means yourself. Okay, and then again, now we have within ID item and the within factor preview. Okay, so then if we ask R to print us out the output here, we've got, so again, we've got a significant effect of preview here. We don't have the sphericity issue here, doesn't really matter. So overall, in this case, we would fairly clearly conclude that we can reject the null hypothesis that preview has no effect on the single fixation duration on N1. Okay. So originally, in 1973, Clark wrote a paper called the language as fixed effect fallacy, where he alerted researchers to the fact that if you are, so language researchers were happily just using repeated measures ANOVAs. So they were just using different words, different sentences, but they did not account for that. They did not include sentence as a random effect in the model. So Clark first alerted psychology researchers and psycholinguistics researchers of the problem that that they were having an independence violation, essentially. So what Clark did was the solution that he proposed was combining F1 and F2 into, so doing the two ANOVAs and then combining F1 and F2 into a joint measure called F-min, and then doing the hypothesis test on the F-min. So, but people soon got tired of this. They argued that F-min was too conservative and just reported separate tests for F1 and F2, basically saying that, well, if I get F1 significant and if I get F2 significant, then I can reject the null hypothesis overall. And similarly, people did T1 and T2 post hoc tests, so pairwise comparisons. So this practice is not strictly correct, but of course it was much superior to not doing anything at all about the second random effect in the design. But now, of course, there's the much more appropriate method of linear mixed models for multiple random effects in one model. And so there is no more excuse for not properly accounting for this. And here the mixed in the mixed models refers to the fact that the models can include both fixed and random effects. Okay, so to start with the linear mixed models, we want to fit, let's try fitting the simplest random effects model possible with LMER. So so here we need the LMER package, the LME4 package, which LMER is part of, and we will I've talked to you about the issue with estimating p-values. So we also need the LMER test package. Okay, and we'll talk about this a little bit later. For now, just keep in mind that, well, basically all you have to do in order to get p-values from linear mixed models is to load the LMER test package. So essentially, that's all you need to do. So let's fit our first model. Let's fit our first model. So we've got, so basically all that we want to do here is to have random intercepts. So remember, this is the model that we talked about earlier, where we just have a random we just have random intercepts. So basically just a main effect of subject and nothing else. So here, this is what that looks like. Here we've, in order to make these easier to compare, again, we are excluding also data points where we don't have an observation for SFDN. But basically, this is what it is. So this part is just like a normal, well, it's just like what you're used to from multiple regression. So dependent variable, then the tilde, and then we've got, here we've got a 1. 1 means that we want the intercept. If you have any other predictors, then you can omit the 1. If you don't have any other predictors, then you have to put in the 1. Okay, so that's the fixed part of the model. So this is basically the, if you remember the formula that I gave you, the mu. And then this part here is saying fit random intercepts for each subject. So this pipe here is sort of saying for. So here, if we look back a couple of slides at our formula for the simplest random effects model, this is the one that's directly after the tilde, right? This is what it fits, this mu. And then the parenthesis 1 pipe or bar subject is this. So this fits random intercepts for each subject, right? Okay. So let's go back to the model. Okay, so that's what it is. And now if we ask R to give us the summary for this, then this is what we get. And you can see we just got the random effects. Actually, this probably looks better in our studio. So I will switch to that because the output was just a little bit cut off there. So, so let's just go down to where we have the simplest the simplest random effects model, right? So let's fit this and do the summary here, and it will print that out here so we can see all of the summary. So you can see it gives us the variance of the subject, which is the when we looked at again, when we looked at the formula for this model. So just switching you back to the presentation for a second. When we looked at this, so this is the variance, right? This is the variance that is estimated here, right? For the intercept. Okay, so let's go back to our studio now. Again, so this is the variance, and then this is the residual variance, which is the epsilon in that formula, right? So so you can see it has two different variance estimates, one for the subject, for the intercept by subject, and one for the residual. And then in fixed effects, it only has the intercept. What does this t-test tell us? Well, it just tests that the intercept is whether the intercept is different from 0, so the estimate. And of course, the intercept is 215.518, so that's very far away from 0. Okay, what's this 215.18? So, well, first of all, this I'm just going to quickly fix this. Okay, so we can take a look if we just do just take these data where we just include. I'm just going to copy and paste that, where we just include cases where we have neither NAs in SFDN1 nor in SFDN, and we just take SFDN1, okay, and we do the mean of that, and we don't need to do NARM equals true because we've already removed the NAs, right? So this is the overall mean that we get, and you can see that is very, very close to the mean that we get, or to the value that we get here for the intercept. Of course, we do have the random effects in there, so that may affect the estimate of the mean a little bit. Okay, let's take a look at what class LMM1 is, just like we looked at the class of LM1, so of the model. So if we just ask R to print the model, then we get a sort of smaller output here. So we just get formula, data, REML criterion at convergence. This is also in here. Yeah, this is also in here. So this is the restricted maximum likelihood criterion that we got. And this is basically, so this was the max, so this is the likelihood that we had when the model converged. Okay, and then we've got, here we've just got the random effect. So here we just got standard deviation, and here we've got the estimates of the fixed effects, and that's it. So it's quite similar to the summary. And if we wanted the plot for that, then we get a fitted by residual type plot. You can see there's, I mean, there really is no, and we don't have any systematic effect fitted. This is just basically, well, the fitted data is just basically predicted by the mean, right? Okay, by the mean and by the random intercepts, right? So that's why you have actually different values predicted here. So all the so you can see all the values, for example, from this subject get the same prediction, but of course the values differ. So that's why you have so the subject does not produce the same value every single time, the same fixation time every single time they make a fixation. So that's why you have these different residuals here. So here for this value, for example, the predicted value was very close to the actual value. Here, the predicted value was very far away from the actual value, right? And again, we're in this model, we are predicting each value just by participant means, basically. Okay. Next model. So so now we are going to add a fixed predictor, and in this case, we're going to add, to keep it simple for now, we're going to add single fixation duration on the preceding word, so SFDN as the predictor. So what we're going to have here is, well, if we call the predictor D, we could also, if we wanted to, we could also call it X. Maybe that would make it a little bit easier. So this is another nice thing about using our studio here. I can basically edit my presentation on the fly. So if we say X here is the single fixation duration on word N, right? So now we have this, we have the random effect of participants, so the random intercepts for participants, and we have a fixed effect of single fixation duration. Okay, and then of course we have the overall error. So let's try this. Let's see what we get if we do this. So this is what this model is. We're calling it LMM2, and let's look at the summary of this model. Okay, here we got it. So so now we have the intercept, and we have the fixed effect of SFDN, and we have, of course, the random effects. Okay, so what has happened here now? Well, we see that the fixed effect of single fixation duration on word N on single fixation duration on word N plus 1 is no longer significant when we include the random effect of subject. Okay, so this suggests that actually that relationship that we saw between single fixation duration on word N and single fixation duration on word N plus 1 in the multiple regression, well, that was really just the product of or the result of these fixations being shared between participants. So, well, of those two coming from the same participant, right? Once we account for the effect of participant by having participant here or having subject here in the random effects, then we no longer have this effect, this fixed effect here. So here we actually had the case of fixed effect not being properly interpretable because we hadn't accounted for a random effect, right? And it makes a lot more sense that what causes two fixation durations from the same person in the same sentence to be related to each other is, of course, that they're coming from the same person, right? Okay. Next up, so these exercises are more for you to do at home to recap what we're talking about. So next thing is we can, apart from looking at the t-test for the coefficient here, we can also do an ANOVA again. Although in this case it's not strictly speaking an ANOVA. Not sure why it's still waiting here. We have the result down here in the console. Strictly speaking, this is not an ANOVA, but a likelihood ratio test, because what we're doing is we're not comparing sums of squares or mean squares like we would do in an ANOVA. Instead, we are comparing likelihoods, and more precisely log likelihoods or deviance. Okay, and what we find is that that the log likelihood of LMM1 and the log likelihood of LMM2, where LMM2 is the one that also has the fixed effect of single fixation duration of word N on word N, those log likelihoods are almost the same, right? They're almost identical. And with this log likelihood, the smaller the log likelihood is, the higher you actually have the likelihood. So adding this one parameter, this slope for n, for single fixation duration on n, only changed the log likelihood, only improved the likelihood by this little bit. The test that you do on that is a chi-square test. So the test that you do to compare likelihoods is a chi-square test, and it is not significant. Okay, so we've got 0.5646, and if we compare that with the in the p value, if we compare that with the p based on the t value here, it's not exactly the same, but it is very close. Okay, now to finish up for today, let's talk about contrasts. Okay, so I'm going to switch back to the presentation for that, but I'll always be just going back and forth. So let's look at the contrast. Oops. So we want, I mean, what we want really, or we wanted to do this entire time, is we wanted to check if we actually have differences between our preview conditions, right? So we don't really care about the relationship between single fixation duration on word n and single fixation duration on word n+1. We really care about the contrasts. So we really care about the levels. And as you remember from the last few lectures on this, when you have categorical predictors, in order to add them to a regression model, as a linear mixed model ultimately is, you have to use contrasts. So today, we're going to use the simplest possible contrast that we can use, which is a treatment contrast. Okay, so before you assign contrasts, make sure that the factor, the variable that you assign the contrast to, is actually a factor. So here I make sure that I make this a factor before I assign the contrast to it. And then I use the contrast function to just assign this contrast treatment. That's the simplest way you can do this in R. Okay, so and then I always like to actually look at the contrasts that were assigned, right? And here the output that I get is that, so for correct is the baseline. So all of the 4 contrasts that are assigned here for the correct condition, they are 0. For the orthographically related condition, the second contrast is 1, all the other ones are 0. For the repeated condition, the third contrast is 1, all the other ones are 0, and so on, right? So the second contrast, the contrast that is called 2 here, compares, it is actually the first contrast that we're looking at, compares orthographic to correct. The one that's labeled 3 here compares repeated to correct. The one that's labeled 4 here compares semantic to correct. And the one that's labeled 5 here compares unrelated to correct. Okay. So let's run the model using the contrast. And once again, the output doesn't quite fit here. So I am going to switch back to our studio. Okay, so here we go. I'm just going to make sure that I assign the contrasts correctly. And here they are. You can see them there. But this is something that R sometimes does, that it doesn't quite get done with the preview. It doesn't matter. We've done it. The output is just fine down here. Okay, and now let's do the linear mixed model where we have preview in here, and we also have random intercepts for subject and for item. Okay, so let's just run this and then look at the preview, the summary for that. Okay, so what do we get? We get a significant t-value for preview 3, and otherwise we don't have anything else. It's interesting. So if you remember, now it would of course be nice if this weren't grayed out, but you remember that the contrast called 3 here compares repeated to correct. Okay, so let's take a look at comparing the coefficients. I'm just going to go back to the slides for that, or at interpreting the coefficients. So the model that we just fitted here is this one. We just have random intercepts here. We don't have slopes. Okay, so we're predicting each observation using the overall with an overall intercept. So that's in the model, that's the fixed effect intercept, right, mu. A random slope for a random intercept for each subject, a random intercept for each item. So that's why we have 2 of the alphas here. And if we look at the model output, we can see that we have the different variance estimates for the intercept by subject and the intercept by item. And then we have these fixed effects for the different dummy contrasts, or the different contrasts. So D1 is the one here that's labeled 2, so correct versus orthographic. D2 is correct versus repeated. D3 is correct versus semantic, and D4 is correct versus unrelated. Okay. So now, when we want to interpret this, so when the preview is identical, so when all the so if we look back at the contrasts, when the preview is identical or correct, right, all of the contrasts are 0. That's the way they are set. Okay, so in this case, we just plug in the 0 for the D's here. Okay, so what it becomes is this. So mu plus A11 plus A12 plus beta times 0 plus beta 2 times 0 plus beta 3 times 0 plus beta 4 times 0. So everything that's multiplied with 0 is, of course, 0, right? So what we get is that in that case, so for that condition, for the identical condition, or the correct preview condition, our observations are just predicted by the overall intercept and the random intercepts, nothing else, and the error, right? When the preview was repeated, so remember here in the repeated case, all the contrasts minus the except for the second are 0. The second one, which is labeled 3 here, is actually 1. So we plug that in here. So we have again mu plus a alpha 1 1 plus alpha 1 2 plus beta 1 times 0 plus beta 2 times 1 plus beta 3 times 0 plus beta 4 times 0 plus the error, right? So we get mu plus the random intercepts plus beta 2. Okay. So now the predicted value is the intercept plus the value of beta 2. Okay, let's go back to our model output to see how that works. Okay. So here's our model output. For some reason, it's still loading in the actual window. But here I can show you the formulas, right? And I can also show you the condition means. This is just a different way of getting the means here. Okay, so in the correct condition here, and what we're doing here actually is we're looking at single fixation duration on word n. So for the pre-boundary words, I'm not quite revealing the result for the critical word SFD N1 yet. I'll do that next week. So here are our fixation times for correct, orthographic, repeated, semantic, unrelated. Okay, and now I'm just going to make this a little bit larger. Now we compare with this our model coefficients, right? So the intercept is 196.564. This is very similar to the correct condition, right? Preview 2 is 4.373. If we take the intercept 1.96 plus 4.373, we get something that is close to 202.49, right? So that is the models, and remember that here, of course, these are the raw the means from the raw data. These are the means that are the model estimate, right? So this plus this is the model prediction for orthographic. This plus this, so 196 plus 8.26 is the model prediction for repeated. This plus this, 1.96 plus 5.589, is the model prediction for semantic. And this plus this, so 196 plus 6.75, is the model prediction for unrelated. And you can see in each of these cases, the model prediction is quite close, right? Okay, so and then for the significance test, what we find here is that so actually the repeated condition is significantly different from the correct condition, because the coefficient here is significantly different from 0. Okay, because we have a p-value of 0.0195. So it does seem to point towards the result that having the, well, even before you're looking at the at the word news, even if you're looking at the preceding word, there is something going on there that that makes people more likely to, or that causes people to stay on that preceding word for a slightly longer time in the case of the repeated word. And maybe, so because this is not the repeated word, this is the word before the repeated word that we're looking at now, maybe it's just that people are noticing something weird, that they have news in the paraphobia, and maybe that causes them to slow down very slightly. The difference is quite small, 8.826 milliseconds, right? But it is significant. Okay, so going back to our presentation, I think I would like to stop it here and leave a little bit of time for questions. Okay, so next week we will talk about the random slopes, because right now we've just been talking about the random intercepts, we'll be talking about using custom contrasts, because actually those contrasts that I've applied here now, the treatment contrasts, are maybe not the best contrasts for my design, because a lot of them test differences that I might not actually care about. Then we're going to talk about how we can use GELMA to fit logistic LMM. So we're going to look at skipping probability in the same data set. And we're going to look at, so ultimately we're going to look at how you can use Bayesian linear mixed model. So how you can use MCMC, Markov chain Monte Carlo methods to fit linear mixed model, so fix Bayesian linear mixed model. So linear mixed models where we don't use maximum likelihood to get the best possible or the best parameter values that we can get, but rather we use Bayesian methods to determine what the to use the data to update our beliefs about what the parameters should be, and then sample from the distribution of those parameters using Markov chain Monte Carlo methods. For this, it may be necessary to install our tools on your computer if you're using a Windows computer. So if you can, before next week, go to this website, cran.r-project.org bin windows/R tools, and follow the instructions there so that you have a working R tools. Because for these Markov chain Monte Carlo models, because they're very computationally intensive, what R actually does, or what the package actually does, is it prepares the model in C code. It then compiles the MCMC model, and it does the sampling, and then it runs the compiled program to do the sampling, just because it takes a long time. Okay, so that is it for today. I think this was a lot of information for you. This video will be available online, so you can go back. And of course, the files are available online, so you can go through the video. You can stop it. I encourage you to run all the code chunks to try everything. And I hope you will get something out of it. I believe you will. Okay, so we have a little bit of time for questions. So please go ahead, put them in the chat. I will just step away for a few minutes to get a glass of water, because I've been talking for almost 3 hours. But please leave me a couple of questions there, and then in the remaining time, I'll answer as many as I can. Okay? So I'm just going to turn off my video for a second while I get some water, and I will be right back. So it looks like we don't have any questions right now. So I can understand that. I can understand that people feel a little bit overwhelmed with all the information I have given you today. So in that case, well, I will be, oh, thank you. Thank you very much. No questions, but you liked it, you got something out of it. Okay, that's great. So in that case, I'll be happy to answer questions next week. For now, I'll leave you with these files. I'll leave you with the video, and then which we will be making available on the website shortly, once it's processed and everything. And And yes, I encourage you to work through it on your own, to be able to pause, because I know I can just keep talking, right? I don't, if no one reminds me to take breaks, or if no one asks me questions, I just keep talking. So but with the video, you have of course, the option to just pause it, think about it, look at the exercises, and see what you can get out of it. And then next week we will complete it with, especially, and I especially want to show you a little bit of how to use Stan and BRMS, because that is something that you don't have a lot of resources about online yet, but it is something that is going to become more and more important for people. And yes, it is perfectly fine to watch it again. I mean, we have 2 sessions. I had to pack a lot into this one, but next week maybe you can make a list of questions that you have. Exactly, you can make a list of questions that you have, and then next week we can go over them, because I realized there's so much information now, and I'm like, okay, come up with questions. Maybe that's yeah, I will give you a little bit of time. In any case, I am very happy that we had almost 100 participants until the very end. So people are just leaving now when I've made it clear that I'm done. So I appreciate that very much. I really hope that it was helpful for you, and that it will be a resource for you in the future. And yes, I'm looking forward to our second session in a week. All right, so I think I'm going to stop things now. I'm going to stop the recording, and I'm going to end the meeting.